Good afternoon, everyone. I'm Dr. Sneha Shinde, and I'll be speaking on gene therapy in thalassemia. So, as we all know, last week Dr. Bilal had presented a study which was published in NEJM. He had presented on gene editing in thalassemia, gene editing by the CRISPR-Cas9 method. So, today I'll be talking on the gene addition trial. So this paper was published in the Lancet in 2024. So the aim of this trial was to achievement of transfusion independence in severe genotype beta thalassemia measure using autologous stem cells and progenitor cells transduced with the lentiviral vector. It was a phase 3 multi-center trial. Age group, the patients included were in the age group of 4 to 33 years, and the total number of patients was 18. So this is an interim result that they have published. Later on in the phase 3 trial, total 41 patients were included, and those reports have been published as abstracts. So the product of intervention was autologous CD34 cell enriched population that contains the HSPCs, that the hematopoietic stem cells, transduced with the lentiviral vector encoding the beta A T87Q globin gene suspended in cryopreservation solution. So, to have an overview of what they actually did, blood stem cells are collected from the patient's body. Then, Zynteglo, which is the product name, it is basically the lentiviral vector transduced with the gene, with the human beta hemoglobin gene. And it is introduced into the stem cells. Later on, this product is then infused intravenously into the patient. So this is somewhat like an autologous stem cell reinfusion. So what is the lentivirus? So it is called BB305 lentiviral vector. It is a replication defective. That is, the virus cannot replicate on itself. Self-inactivating third generation HIV-1 based lentiviral vector carrying the human beta globin gene with a single modification at codon 87. So this lentiviral vector is used for ex vivo transduction of the autologous stem cells that have been collected. So what the lentiviral vector does is it integrates a functional beta T87Q beta globin gene into the patient's stem cells, and their erythroid progeny produce the beta A T87Q globin, forming the functional hemoglobin in circulating RBCs. So the virus, the lentiviral vector, is basically, it is a shell, or it is like a delivery machinery carrying the RNA encoding the engineered beta globin expression cassette. So inclusion criteria, they included patients less than 50 years. The patient had to be a transfusion-dependent thalassemia phenotype, that is at least getting more than 100 ml per kg per year.
Sneha, if I may interrupt, what was the lower age of inclusion?
Lower age, ma'am, the median age was 12 years and the lowest age was 4 years. 4 years. They should have a good performance status with Karnofsky more than 80. A detailed transfusion chelation records. So for the last 2 years, the patient should have received transfusion at a comprehensive care center with proper transfusion records, chelation records, all other organ function records. So this was an important inclusion criteria. Exclusion criteria was presence of other mutations like other than the beta 0 mutations. So beta plus, beta E, beta C on at least one HBB allele. Any viral markers should be negative, and other criteria were similar to the previous study. So it is an extensive list of exclusion criteria which mainly pertains to infections, active infections, immunosuppressions, suppressant steroids, or any autoimmune diseases. So it's an extensive exclusion criteria. So the primary endpoint of the trial was transfusion independence, that is an HB of 9 grams per deciliter without transfusion for the next consecutive 12 months. And the secondary endpoints were proportion of patients with transfusion independence at month 24, duration of transfusion independence. So they also wanted to look at the sustainability of the product, time from infusion of the Beticel, that is the product, to meeting of transfusion independence. How long does the patient require to achieve transfusion independence, and the weighed average hemoglobin during transfusion independence. So moving on, what they actually did. So after a proper rigorous screening of the patients as per the inclusion and exclusion criteria, mobilization and apheresis of the patients was done. So mobilization including GCSF. So day 1 to most of the patients received for 5 days, and plerixafor they received for around 3 days. And the apheresis was done for 3 days. So day 1, day 2 of the apheresis was mainly for collecting the stem cells required for the actual infusion, and the third day was reserved for a backup stem cells. So in case there was any issue with the product manufacturing, delivery, transport, then you had the stem cells backed up. Myeloablation. So 4 days of myeloablation was done. So in children they used busulfan at the dose of 0.8 mg per kg. It was a 2-hour infusion given every 6 hours, whereas in adults it was a slightly different dose, 3.2 mg per kg once daily, and it was a 3-hour infusion. So as we all know, with busulfan we need to see the levels, whether they have been achieved in this the serum levels, whether they have been achieved or not, and accordingly titrate the doses. So in children the levels were collected at the end of the first 2-hour infusion, and after that, after every 15 minutes, after 30 minutes, and accordingly around 6 times the levels were collected. Can I go ahead? Next step was the infusion. So infusion, the dose, the median dose that they targeted was 5 into 10 raised to 6 per kg CD34 cells. After that, the patient was observed and discharge was done after neutrophil engraftment. That is, at least 500 ANC should be seen in the patient's CBC. After that, there was a strict follow-up schedule. So in the first year, they were following up the patient every monthly. And after that, in the second year, every 2 monthly. In the third year, every 3 monthly. And after that, 6 monthly. So this is the criteria that has to be met prior to the myeloablative conditioning. So there are certain criteria that they put up. So mainly, like in other gene therapy trials also, hypertransfusion was advised more than 30 days prior to the conditioning to maintain an HB of 11. So usually we maintain it around 9 to 11, but here the patient who are included in the trial, they had to maintain an HB of at least 11. Iron chelation therapy has to be stopped more than 7 days prior to start of conditioning, but deferasirox patients who were on deferasirox, they had to stop it 30 days prior to the start of conditioning, and in place of deferasirox they can use the kelferol or desferal. Third thing was use of prophylaxis to counteract the veno-occlusive disease. Then for busulfan anti-seizure prophylaxis, so we usually use clonazepam, but here they used phenytoin.
We use now, what is the new drug?
Leverastat.
Leverastat.
Okay,
Leverastat.
we have used clonazepam.
They were using
in
Leverastat. CTC, we were using Leverastat.
So
phenytoin is very difficult.
Yeah, but they have
drug
used
interactions are so
Exactly.
many.
Interactions
And side effects also.
liver
So,
side effects,
yeah.
and even
And because
Effects
you're worried
can be
about
very-
VOD,
especially in children, we have
in
to be
children.
more careful. So the product to be used for a particular patient had to be released, tested, had to be dispositioned for clinical use, and it is stored at the clinical site. After that only the patient can be taken up for myeloablation. And the patient has undergone adverse event and concomitant medication assessment. So as we discussed, chelation, physical assessment, everything has to be assessed properly before taking the patient in for myeloablation. So infusion, the product infusion can be done more than 48 hours post the conditioning, and after thawing at 37 degrees Celsius, it has to be infused within 4 hours. Dose is equal to or more than 5 into 10 raised to 6 per kg, and the monitoring during and after transfusion has to be every 30 minutes for the next 2 hours. Post infusion follow up and discharge. So as mentioned before, discharge was done after neutrophil engraftment. Follow up monthly for the first 12 months, then 2 monthly for the next year. So this was another study, like a follow up study called the LTF 303. So here they aim to follow up the patients for the next 15 years. So coming to the results, total 18. So in this interim results that they have posted, they have evaluated 18 patients who were infused. Total pediatric population, that is less than 12, was 8. Adolescent 12 to 18 were 5 patients, and more than 18 were 5 patients. Median duration of hospital stay was 42 days. 89%, that is 16 out of the 18 patients, achieved transfusion independence as per the primary endpoint. Time to achieve transfusion independence, median time was around 15.7 months. Median HB during the transfusion independence was 10.5, and most patients were able to discontinue the iron chelations. So as we can see in this graph over here, the x-axis denotes the months post infusion, and the y-axis is telling us about the hemoglobin. So at third month, and we can see the red is for the new HB, that is the modified HB that has been which was supposed to be activated in the patients, and the blue line is for the total hemoglobin. So as we can see, as time went ahead, there is sustained response. So the HB is maintained around 10, and even the newer HB, which is the functional hemoglobin in these patients, that is also sustained. So this was, so basically the primary endpoint of the trial of the study
Just
was
one
achieved.
question,
Yes, ma'am.
Sneha. Time to reach transfusion independence was 15.7 months as the median.
Median.
What was the range? If you can remember. If you don't, we can see it later.
Ma'am, I think.
Means the earliest was.
Yes, ma'am. I think I have a graph in the
you're going
subsequent
to show.
slides.
Okay.
So this is the drug product characteristics. So percentage of lentiviral vector positive cells, that is the transduction happened in how many cells? So 78% of the stem cells were transduced with the lentiviral vector, and the dose which was infused was 10.8. That is the median dose, starting from 5.9 to 42.1. So it's quite high. Mobilization details. So the pre-decided stem cell yield that we wanted was 16 into 10 raised to 6, which is also quite high compared to autologous transplants, where we expect around 3 to 4.
Even 2 is good enough.
Yes,
sometimes.
So
depending
those
on how heavily pre-treated they are,
and age of the patient,
age of
everything will not.
So up to 5 is good, but anything below 2 is not acceptable.
So most of the patients, 78% of the patients could get done only in one cycle, whereas there were 22% patients who required 2 cycles, and I think 2 to 3 patients required even the third cycle. GCSF Plerixafor were given in almost all patients. Number of apheresis collection days per mobilization, so it is around 2. And number of stem cells collected was median was 30.9. So they had a good yield considering everyone received.
Just another question. You said 3 Plerixafor doses, 3 days you gave. Was it 3 days or 3 doses?
It was 3 days, ma'am.
And every day single dose?
Single dose.
Not 12-hourly?
No, it was every day 24-hourly dose. So coming to the engraftment, neutrophil and platelet engraftment occurred at a median age of 26 days and 49.5 days. So I reviewed a few more studies in which they have concluded that the splenic function of the patient and the spleen size, it had significantly affected the platelet engraftment. So here we can see platelet engraftment took almost 1.5 months. So which is significantly high. I think we discussed this point in the previous, during Bilal sir's this also presentation also. So
only there one point which Amit had made, if you remember, that if you use the fluid trio protocol, you might reduce the engraftment time.
Yeah, and the number of hospital days also you can
reduce. So maybe that should be also reviewed in detail, when we probably take up gene therapy as a routine.
So moving ahead. So this is the median duration of transfusion independence, ma'am. Like you asked. So here the x-axis is telling us about the months after the drug product infusion, and this is the age. So as we can see, the red dots are the PRBC transfusions.
Okay.
The orange lines are the patients who achieved transfusion independence, and these 2 patients could not achieve transfusion
So there are only
independence.
2
Only 2.
outliers, right?
Yes.
That is what I was concerned when you said the median was about what 15.4.
Ma'am, they have not achieved transfusion independence.
So these are failures.
Failures.
But amongst those who achieved, you said how much time it took? 15.4 months.
Yes, ma'am.
Was the maximum?
Yes.
Or was
that
the
was
median?
the median. That was the median, ma'am. So here, as we can see, so the red dots are the PRBC transfusions.
15.4 months or days?
It is months, ma'am, because
Months after DP infusion, if you see here, 0 to 12 either hai, so you are almost all are becoming independent soon after, na?
Ma'am, but the HB that they need to achieve, that is, so the HB they targeted for transfusion independence, the definition was 9. So maybe they were not maintaining
so they
the
require transfusions only here, na?
Yes.
These red dots.
Yes.
So some of them, they are very quick, within a month, probably. So I think it must be 15.7 days, Sneha, because they are not supposed to go on for 15 months with transfusions.
Ma'am, I'll have a look, but I think what they mean is the HB to be achieved was 9. The
Yeah,
stable
yeah,
HB for
9.
12 consecutive months.
That
The
is-
definition of transfusion independence was HB of 9 or more
Yeah,
than 9 for
understandable.
12 consecutive months.
That those are the ones who will not transfuse.
Yes, ma'am.
Within 3 months, that means they have become transfusion independent, and then they have observed those cohort who have become
No, no,
transfusion.
but she said median time to transfusion independence was 15.7 months.
But does that include the
Ma'am, I'll
12
still
months?
go back to the. I'll
Okay,
go
I
back to
want
the
you to
study
check.
and
If you
clarify. Yes, ma'am.
have the study, I can go through while you are presenting on the laptop only. Okay. We'll see later. But
Yes, ma'am. We will have a look at the point. So this is the list of the adverse events that were seen during the trial. So the first part tells us about the grade 3 or more severity of adverse events. So thrombocytopenia, neutropenia, anemia, FN. So these were quite expected and manageable adverse events. The most common was cytopenias and gastrointestinal disturbances. Most severe was pyrexia. And fortunately, there was no GVHD, insertional oncogenesis, or any deaths till date.
then how much follow up you said?
Ma'am, I'm going to talk about the long term.
So insertional oncogenesis, they have looked in the
Yes.
NTFU wala
Yes.
cohort, na?
Yes, ma'am. So just to have an idea what the lentiviral vector cells and the drug characteristics, how did they affect the outcomes? So percentage of the lentiviral vector positivity in the drug product for 2 patients who did not achieve, like we saw in the previous graph, they did not achieve the transfusion independence. So the transduction was lower than the median value. So it was only 61 and 57%, whereas the median was 79.5%. And a recent analysis of the beta cell phase 3 data has shown that most reliable predictor of transfusion independence was the percentage of the lentiviral vector positive cells in the drug product. So, I mean, it is very logical and it makes sense. Recent improvements in the manufacturing process of the lentiglobin BB305 drug product have led to higher percentage of transduced cells and the vector copy numbers in the in vitro and to higher VCN in the bone marrow of mice in non-clinical studies. So in phase 3 only, the initial product was called LG207 and the newer product is LG212. So in 212, they could see that there was a better transduction of the stem cells and more patients were transfusion independent when they received the optimized product. So long-term follow-up. So till now, we have a follow-up of around 5 to 6 years. So total 63 patients have received the infusion. Out of that, we have 10-year follow-up of 2 patients and 5-year follow-up of 51 patients. 41 have received the commercial, that is the optimized, that is the LG212 product. And out of the 41 patients, 37, that is around 92% have achieved transfusion independence. 19% of the total cohort had side effects. 5 patients had a VOD in spite of giving the prophylaxis, and all 5 received defibrotide and have recovered. So there were no deaths till now. No malignancies, insertional oncogenesis, or vector delivered replication competent lentivirus were reported. So the lentivirus was safe. Serum ferritin and liver iron content as per dry weight both have reduced significantly. And even patient reported outcomes like quality of life, mental and physical health as per the SF36 and PEDS-QL scoring, they are also much better. So we don't have the long term follow up as a complete study published. We have a small abstract, so I have quoted from that. So highlights comparing the Beticel, that is Zynteglo, with Casgevy. So the core strategy, that is Beticel used gene addition. So it added a lentiviral vector carrying the BAT87Q globin gene. So it was a modified beta globin gene which was inserted into the lentivirus and then into the stem cells. Whereas Exaccel, the NEJM study, it used gene editing, that is ex vivo CRISPR-Cas9 induced gene editing, and it targeted the erythroid enhancer of BCL11A. So it targeted the BCL11A gene, and then it raised the HBF. So the mechanism of action of both the modality, both the gene therapies were different. So in Zynteglo, the adult hemoglobin with beta, the modified hemoglobin was increased, and that is how it led to transfusion independence. Whereas in Exaccel, the gene editing, we have increased the HBF, and hence caused transfusion independence. And in phase 3 studies, the efficacy is some more or less same, around 9. It's comparable, definitely. In both phase 3 studies, had more than 90% of the patients who achieved transfusion independence and also a sustained response. Adverse effect profile was quite similar, mainly hematological, GI, VOD, and transaminitis. Thank you, everyone.
Go to